Thanks very much, Claire. Hope you can hear me. And
Very well, thank you.
Great. Good afternoon, everybody. So I'm going to talk about our two sessions, which are on the determination of concentrations of nanoparticles in biological samples using single particle ICP-MS. So if we could have the next slide, please. So the overall objective was really to identify any additional data and also to consider priority areas to focus on to help us develop single particle ICP-MS as a robust measurement approach for a range of engineered nanomaterials and more importantly, biological sample matrices to support eco and toxicology studies. So we had two sessions. The agenda for each was broadly similar. We had a welcome with objectives, then a sort of thought starter presentation, a discussion with focus questions, and then followed by a summary. Although there was so overlap between the two, we had a different focus for each of the sessions. So the first session was on extraction protocols, and these are how effectively we get from nanoparticles within a biological matrix to nanoparticles within a solution that we can introduce to the equipment which will measure the size and the concentration of the nanoparticles. So for the first session on extraction protocols, we were looking at focusing our questions for discussion around existing data. What are the data gaps? Where would it be useful for us to undertake some new experimental studies, and also whether or not we could make recommendations about which extraction protocols are the sort of most appropriate in particular circumstances. And the focus for the second session was really on measurement and validation, which is an important question because really we want to ensure that the results that we're getting from using these techniques are an accurate reflection of the reality as they can be. And in the absence of certified reference materials, this is a sort of a bit of an ask sometimes. So if I could have the next slide, please. So for session A, this is where we're focusing on our extraction protocols. We started really by looking at a literature review that we'd undertaken, looking at what's already been done in terms of different ways of extraction protocols used with different biological samples and different approaches people have already taken out there to give us a background of what information is there. And we had a wide-ranging discussion around this, which raised a lot of questions, a lot of issues and areas where we could do some, we could usefully provide some more guidance. So if I could have the next slide, please. So these are just some of the questions. I don't want to go through them all in detail, but there were questions around sample storage procedures, even before we start the analysis, whether or not extraction protocols affect the particle size distribution, or do they even introduce new particles, they create new particles, and whether or not we need to validate against alternative methods, and also issues with detection limits. So we had a lot of questions, and we will be looking forward to sort of diving into these more in detail in future. So if I could have the next slide, please. So one of the things that we wanted to look at was data gaps. And again, we focused really on our original literature review. And this is just really a summary of what's out there in terms of analysis of different tissue type, different biological matrices, plants, mammalian fluids, and other creatures, and also the nanoparticles that have been studied. And we had some additional suggestions shown in red on the left of different tissues we could be looking at, perhaps bone and some of the biofluids for ecotox, and also a range of additional nanoparticles that people considered might be a useful further set to consider possibly in future experimental studies. So that was what we focused on for session A. If I could have the next slide, please. Sorry about that. So for session B, as I mentioned, this was focused very much on validation. And we sort of went through 4 questions. The first one was really, in the absence of a certified reference material, is the spike recovery approach the most useful to determine recovery? And I think the answer to that generally was yes, but we also need to consider other approaches, such as to ensure that we sort of provide convincing evidence for the results. Things like mass balance, whether or not it be useful to do round robins to build consensus, and also comparison with other techniques. But also people raised some of the potential issues that, for example, spike recovery can depend on the type and mass of the spike added. How do we demonstrate complete digestion? And issues such as the fact that biologically incorporated nanoparticles are typically harder to extract than the spiked nanoparticles. So a range of interesting issues raised there. The second question we looked at, what are appropriate criteria for acceptance analysis, for acceptance of the results of analysis? And we started discussing that really from the sort of comparison with the use of certified reference materials for, say, metal extraction using ICP-MS, where something sort of up to a 20% difference might be considered appropriate. And I think generally the feeling was that we shouldn't perhaps focus too much on very strict levels, because these are complex analytical approaches, and also they were very the importance of such levels varies depending on the purpose, what the aim of the use of the result is. So that was a very useful way forward, and I think it's a sort of case by case basis. One of the questions, the third question, was on what can we learn from other areas. We didn't really get much response in that area. I think it was quite a complex situation to try and cover such a wide range of other sort of areas. But the final question we had a very long and very useful discussion on, and that is what would be an appropriate roadmap for the development of certified reference materials. So we discussed various options, for example, growing cells with nanoparticles using cancer tissue models. And we also wondered whether some current certified reference materials may actually contain nanoparticles, and whether it would be worthwhile investigating them using various databases. And we also talked about perhaps funding mechanisms, the need to demonstrate potential demand to convince manufacturers, and also that you'd need to involve producers that were accredited to the correct ISO standard. So a lot to think about in terms of validation. So if I could have the next slide, please. And before I get to the final comments, I just wanted to thank all the participants for their contributions, both during the sessions, and we had some information provided before and since the workshops, and this has all been very helpful. So next slide, please. So just some final points. So the implications for the project, I think the general feeling is that there was general support for the approach we've taken so far, but we had some very interesting issues raised and suggestions on ways forward, and also clearly a recognition of the need to provide more guidance in specific areas, and especially on these validation approaches. In terms of next steps, clearly what I've given you is a very rushed sort of introduction of some of the key points, and what we need to do is go back and fully digest the workshop discussions from the chat and the recordings, and report back at the NanoHarmony webinar on the 16th of December. On a longer timescale, we need to complete the data collection and gap analysis document, which will inform us on sort of prioritize our way forward, and also develop plans for future activities, including experimental studies, to address the key issues and priorities identified here. So we're very grateful for everybody's input, and if you have any additional comments and suggestions, please do email them to Richard or myself, and we'll be very interested to get more responses on the way forward. And apologies for this rush through, but 10 minutes really doesn't quite give you enough time to do justice to an extensive 3-hour discussion. But thank